Bonjour tout le monde. Going to shift to English because we have an American guest today. It's a great honor to welcome and introduce Professor Andy Miller. You all know that probably that he's professor and vice chair for research in the Department of Psychiatry at Emory University in Atlanta in the States. And you all know that he is an international expert in the field of immunopsychiatry. He's been extensively studying the relationship between brain and immune system. And as we are making a survey of all innovation in psychiatry during this year conference, it's extremely important to mention that he has conducted the first clinical trial studying the effect of immunomodulator and cytokine antagonist in the treatment of depression. So it's really a way to see the links between academic research and innovation and how we can use these new tools to better diagnose and treat our patients. So Andy, thank you very, very much in a very busy day, I know, to spend some time with us to explain to this group of young scientists and clinicians the field of immunopsychiatry. So the idea is that you take your time to explain and teach us, and then we have a short-term discussion. So don't hesitate to ask questions even in French, either in the chat or after the talk, and I will try to translate the questions. Andy, the floor is yours. Thank you very much.
Great. Thank you so much, Marion. This is a great honor for me to have this opportunity to speak to you all and share with you some of the work that we've been doing on the effects of inflammation on the brain and how this can be applied for developing new treatments, and particularly targeted treatments, targeting the immune system to treat psychiatric disorders. And so that's going to be my focus this morning. Perfect. Just before I begin, a couple of disclosures. I'm a consultant for Cerevel Therapeutics as well as Boehringer Ingelheim. I like to start maybe a talk like this with a quote from Martin Luther King Jr., who I'm sure you're all familiar with, from the United States, civil rights activist, and to emphasize that we as mental health researchers really need to embrace this notion of the fierce urgency of now. What we need to take our extensive knowledge, and we have a lot of knowledge about how the immune system affects the brain in this case, and apply that knowledge to ultimately change people's lives. Life's most persistent and urgent question is, what are you doing for others? And that is our most urgent agenda. Fortunately, under current circumstances, as we're going to see today, I'm afraid we're not going to get there. And so we're going to have to make some changes. We're going to have to do things differently. And that said, today I'm going to walk you through some of the issues that are plaguing the development of targeted treatments in this space, the brain immune space, and discuss some possible solutions. So to put this in context, I'm gonna start with depression, which is where much of our work and the work of immunopsychiatry has focused. And I think most of you are aware that depression is a devastating disease. It is, in the United States, at least just under 10% of the US adult population is afflicted with depression. It is the 10th leading cause of death by suicide in the United States, and it is the leading cause of disability worldwide. The problem, however, is, and above and beyond the scope of the disease, is the fact that for those of us who treat depressed patients, we're faced with the vexing problem that about a third of depressed patients don't respond to our conventional antidepressant therapies. And that includes not only medications, but also psychotherapy. And this kind of emphasizes this urgent need for us to do a better job, to think outside the box, to come up with new treatments, to ultimately improve outcome for patients with depression, especially these individuals who are dealing with treatment non-response. So that's where the sort of the immune system comes into the equation. The immune system being a novel mechanism by which patients may come to their depressions, and there's quite a bit of data. I know that some of you may be very familiar with this literature, but there's quite a bit of data to suggest that inflammation plays an important role in the development of depression. There's quite a bit of data in this area. Patients with depression exhibit all the cardinal features of an inflammatory response, with increases in inflammatory cytokines. There have been hundreds, if not thousands, of papers that have been published in this area. Interleukin-6 and tumor necrosis factor are the most reliably elevated in this regard in meta-analyses of this literature. There are also increases in acute phase reactants or acute phase proteins. These are produced by the liver in response to inflammatory cytokines, play a role in containing the inflammatory response, help clear pathogens, help mobilize the wound healing process. And C-reactive protein, or CRP, is the most reliably elevated in meta-analyses of that literature. And then there are also increases in chemokines, which draw cells into the site of inflammation, cellular adhesion molecules, which make cells stick at the site of inflammation. And these two have been found to be elevated in depressed patients versus controls. And you can go into the brain, postmortem brain samples from depressed patients who've committed suicide, and in the brain parenchyma, you'll see evidence of activation of inflammatory signaling pathways in the brain, as well as activation of the immune cells of the brain, the microglia, as well as evidence of immune cell trafficking to the brain. And interestingly, it appears that there's a special relationship between inflammation and treatment non-response, such that if you have increased inflammatory markers prior to treatment, you're less likely to respond to conventional antidepressant therapies. It's been shown most extensively with serotonin reuptake inhibitors. And if you've failed multiple treatment trials, you're more likely to exhibit an increased inflammatory response. And this is just an example of this from a paper we published several years ago. There's just under 100 depressed individuals, all unmedicated at the time. And we asked them, in your current episode of depression, how many treatment trials have you failed? And you can see that the majority of the patients had not been treated at all in their current episode. But of those who had failed 1, 2, or 3 or more treatment trials, you see this stepwise increase in TNF as well as IL-6. Same thing with the soluble receptor for TNF. And you see a similar pattern with CRP. This trend here is significant, although we didn't see any significant differences between groups, although the UK group has demonstrated that treatment-resistant depressed patients have increased CRP compared to treatment-responsive depressed patients, which we didn't have in this study, as well as healthy controls that were also not included in this study. The second major body of data that suggests that there's this link between inflammation and depression, and it kind of gets to the cause and effect nature of this relationship, is you can give an inflammatory stimulus, inflammatory cytokines. I'll be talking about interferon alpha, which is an inflammatory cytokine that causes depressive symptoms, 30 to 50% of patients, depending on the dose. Other inflammatory stimuli, endotoxin, typhoid vaccination, typically given to healthy volunteers. And these stimuli cause depressive symptoms. We've learned a lot about the neurocircuits that are involved and the neurotransmitter systems that are involved in the effects of inflammation on the brain based on this work. And finally, as you might guess, that you can inhibit inflammation. Again, addressing this cause and effect, you can inhibit inflammation, and that reduces depressive symptoms. And this has been shown most convincingly in individuals with autoimmune and inflammatory disorders who are given anti-cytokine therapies, which have then been shown to decrease depressive symptoms. But then the question becomes, well, what about the efficacy of anti-inflammatory drugs in otherwise healthy depressed subjects? Obviously, a question that is of quite a bit of interest to us. And there have been a number of studies that have looked at this using a variety of different anti-inflammatory agents. And there is large enough literature that a number of meta-analyses have been done. And these demonstrate that, yes, there is a modest effect of anti-inflammatory drugs in treating depression. However, the results have really been underwhelming. And investigators have asked, is there really anything there? And unfortunately, to compound things, is the fact that two of the largest studies to date, which were both done very recently with subject numbers 60 and above in the various treatment groups, these studies use two of the major drugs that have been used in these previous meta-analyses, celecoxib, minocycline, and both of these studies found no separation whatsoever between the anti-inflammatory drug and placebo. And this is just an example of one of these trials, minocycline and celecoxib. Alan Young, the senior author here. And as you can see in the conclusion here, no evidence that minocycline or celecoxib was superior to placebo. And this is the conclusion, which is extremely disturbing and puts the field in great jeopardy. This large trial casts doubt on the potential therapeutic benefits of anti-inflammatory drugs for the management of depression, bipolar depression in this case. That's a problem. If you're someone like me who's trying to develop drugs that are targeting inflammation, when you see papers like this, the funding agencies say, hey, well, what about that paper? You're going to just make us have us spend more money on something there's no there. And so this really poses a significant threat to the field. Now, why might these trials be underperforming? And so this really gets to trial design and how these trials have been conducted. And the biggest problem, or of the many problems, but one of the biggest ones, is that the sample has included invariably for these studies, maybe a handful have not, but there have been hundreds of these studies, have typically used heterogeneous depressed patient populations not enriched for increased inflammation. As we'll talk about in a minute, only a subgroup of patients show increased inflammation. So if you just take a bunch of depressed patients, you're very unlikely to see a signal in this small group of patients who have increased inflammation. The second problem, which is a problem for many clinical trials as we try to develop targeted therapies in psychiatry, is the primary outcome variables are very nonspecific. We're all using Hamilton depression scales, for example, in depression, and we're not really looking at those outcome variables that are specifically related to inflammation. We're going to talk a lot about that today. What are the specific things that inflammation does to your brain that would then make sense for us to use as primary outcome variables, as targets for treatment? And finally, the drugs that have been used are really not terribly good anti-inflammatory drugs because they have multiple off-target effects. And the studies have not even measured whether or not the drug they're giving is reducing inflammation in the first place, nor looking at the neurocircuits and the behavior that inflammation specifically affects. So again, that relates somewhat to these design issues. So let's talk a little bit about what we can do, what might be some principles for developing therapies, targeted therapies, in this case for inflammation. And this, what I'm going to talk about today, is it really applies to any type of targeted treatment development in the context of understanding specific mechanisms that are causing psychiatric disorders. Well, one of the first principles is we need to enroll patient populations that are enriched for inflammation. That's pretty obvious, should be pretty obvious to all of you, and we can talk a little bit about how that can be done. And secondly, we should be focusing on targets and treatments that are really based on what we know about how inflammation affects the brain and behavior. And so we'll spend some time talking about both of these issues. Now, what about enrolling patient populations enriched for inflammation? Well, the first thing to recognize is one size doesn't fit all when you're developing targeted treatments. Most of the treatments that we use in psychiatry are interchangeable. You have a depressed patient, you can treat them with an antidepressant, an antipsychotic, a mood stabilizer. It really doesn't matter. These drugs are highly nonspecific and work across disorders. This is not the case in inflammation. Depression is not an inflammatory disorder. In fact, inflammation is only increased, as I mentioned, in a subgroup of depressed patients, and it's about 30% of the depressed patients, depending on the sample. Now, I was talking before about mean differences between groups, but those mean differences in inflammatory mediators between depressed and controls are being carried by the 30% that have the increased inflammation. I should also mention that inflammation is also increased in a number of other disorders in association with a similar behavioral changes. And this is the case for mood disorders, anxiety disorders, schizophrenia, neurodegenerative disorders, medical illnesses, all where we see evidence of increased inflammation in association with changes in specific symptom domains. So what we understand is that inflammation's effects on behavior are not about any specific disorder. This is truly a transdiagnostic phenomena, which makes this a challenge as to, well, what patient population do you look at, or is this something we should come up with a new disorder altogether? And also, if there's significant inflammation, regardless of the disorder, it's going to have effects on specific neurotransmitter systems, specific neurocircuits, and related symptoms, and this will occur across disorders. And we'll get into a little bit of this as we move through my presentation today. So who are the people that have increased inflammation? They're all on this slide here. You can select patients with increased inflammation based on a lot of phenotypic characteristics. Probably the most common that I see in my practice are these first two: patients who are maybe the first three, patients with obesity. Fat cells grow very, very quickly. They outstrip their oxygen supply. The cells die. And that, as far as the immune system is concerned, is a wound. And so macrophages, other immune cells will rush into fatty tissue, and this tissue here is loaded with macrophages that will produce inflammatory mediators and change the metabolic processing in the body that can contribute to obesity. And obviously those inflammatory mediators can get into the brain. We'll talk more about that later. As people get older, they show many of the same metabolic changes that occur in obesity that drive inflammation. And so there's a process of inflammaging. So as people get older, they start to exhibit increased propensity to inflammatory responses. And actually, we're seeing older individuals developing de novo autoimmune and inflammatory disorders. Early life stress can change neuroendocrine set points that regulate the immune system early in life, and in combination with chronic stress can trigger an inflammatory response. A lot of talk about the gut microbiome and dysbiosis, where microbial products from the gut can get into the circulation and activate the immune cells in the inflammatory response. Many of the common chronic illnesses that people are dealing with are associated with inflammation, as are chronic infections. And then, of course, in the medical setting, there are many things that we do to patients: surgery, radiation, chemotherapy, even immunotherapy, that can lead to chronic persistent inflammation in these patient populations. Now, of course, you can select patient populations, but it's always nice to have a biomarker. Can I do a blood test and find out whether somebody's inflamed or not? And we've advocated for a C-reactive protein, or CRP, being a good enough biomarker of inflammation. And the reason we believe that is this is a marker of inflammation that is immediately available, at least in the United States, in virtually every clinic and hospital. We even use it in our research setting with a point of care testing, where you do a finger stick get a little bit of a drop of blood and put it into a machine. We know immediately whether somebody has high, medium, or low inflammation. CRP is run under standardized protocols in the United States using turbidimetric assays. So you get reproducible results across laboratories. So your CRP in New York is going to be the same as your CRP in California. And this CRP has been used extensively in other medical disciplines as a way to measure inflammation. And this has been used extensively, especially in the cardiovascular, in the area of cardiovascular disease, and is now being used more in metabolic disorders as well as in cancer. Now, we like CRP for some more scientific reasons as well. And that's that CRP maps nicely onto a number of other inflammatory mediators. And you can see here, these are popular pro-inflammatory cytokines and their receptors. And this is a measure, a composite of all these inflammatory mediators. And if you look at a plasma CRP greater than 3, which thanks to the American Heart Association and the Centers for Disease Control and Prevention in the United States has been deemed as high inflammation. If you have a CRP, plasma CRP greater than 3, look at the composite of these inflammatory mediators. Nice increase here compared to individuals with lower inflammation, measures of inflammation. And if you look at the individual cytokines, you can see pretty much across the board, they're all elevated compared to individuals with lower levels of CRP. So it's a good marker for, in general, other inflammatory mediators that you might measure in the blood. What's interesting here is this is not only relevant to what's going on in other inflammatory mediators in the blood, but you can go into the brain and get cerebrospinal fluid and look at that same composite of inflammatory mediators. These are patients with high inflammation that you measured in the plasma, and look at the composite for the inflammatory mediators in the brain, in the cerebrospinal fluid, in the CSF. And you see, again, this very marked separation between those with high plasma CRP versus those with low. And you see pretty much not quite as good as what you saw in the plasma, but pretty much across the board, you see elevations in these inflammatory markers in the high CRP group versus the low CRP group. So CRP is a good start to identify who in your patient population has high CRP and would be a good candidate for a targeted treatment that is going after inflammation. And as I mentioned, none of the previous studies, or maybe a handful of previous studies, have used CRP or other inflammatory markers to separate out the populations of patients. So what about the targets and the treatments? What should we be looking at in terms of the effects of inflammation on the brain? What treatments are available to us to develop targeted treatments that are addressing this pathway from inflammation to changes in behavior? This is my map of inflammation's effects on the brain. We know a ton about how inflammation affects the brain through studies in cell culture, through laboratory animal studies, as well as studies in humans. We know that inflammation affects neurotransmitter metabolism, influencing monoamines by decreasing the synthesis, decreasing the release, and increasing the reuptake. So we get a decrease in monoamine availability, which is a cornerstone of the monoamine hypothesis of depression. Inflammation also influences the function of astrocytes and their regulation of glutamate. So you see astrocytes show increased release and decreased reuptake of glutamate, and the glutamate will build up in the synapse and spill out of the synapse, this extrasynaptic spillover, and actually activate adjacent neurons, leading to chaotic signaling locally, and that can disrupt functional connectivity in circuits of the brain. And also, inflammation can affect growth factors, neurogenesis, and ultimately decreasing neuroplasticity in the brain. And these effects on neurotransmitter metabolism and neuroplasticity ultimately affect the circuitry in the brain. And we've learned a lot about what circuits are engaged by inflammation in the brain, largely through studies where individuals have been administered inflammatory stimuli. And there appear to be two major circuits that are reliably engaged by inflammation. one circuit that's involved in anxiety, arousal, and alarm, and involves the dorsal anterior cingulate cortex, insula, hippocampus, and amygdala. And the other circuit is a circuit that's involving the basal ganglia, in particular the ventral striatum and the dorsal striatum. We'll talk more about this in just a minute. And its connectivity to the ventromedial prefrontal cortex and the subgenual anterior cingulate cortex. And this circuit here is involved in motivation, as well as some pathways going up into the motor circuits here. So the motivation and motor activity. And if disruption of this circuit here leads to loss of motivation, loss of interest of pleasure, and the symptom of anhedonia, which in my opinion is the most devastating symptom in depression, and it's seen in multiple other psychiatric disorders. So when we think about targeting the effects of inflammation on the brain and behavior, maybe anhedonia is the symptom that we should be focusing on, and not depression writ large. Based on the fact we've done some work here on this anxiety circuit, I'm not going to talk about much of that today. I'm going to focus here on this circuit here where we've done most of our work and talk a little bit more in detail about the impact of inflammation on these motivational circuits. Now, to study this, our lab has seized upon the opportunity of patients undergoing treatment with interferon alpha, which, as I mentioned, is an inflammatory cytokine that leads to depression in 30 to 50% of individuals, depending on the dose. The cancer patients who are treated with interferon alpha, it was actually one of the early immunotherapies. They're the ones with the higher rates of depression, and then infectious diseases like hepatitis C, lower rates of depression, but virtually everyone develops a neurovegetative syndrome that has a lot of anhedonia and fatigue and loss of motivation involved. And so we wanted to understand, well, what's going on in the brain in individuals who are receiving interferon alpha? So in order to do this, we looked at the ventral striatum and its activation in the context of a hedonic reward task using functional magnetic resonance imaging. And here you can see this is the area of the ventral striatum. It includes the nucleus accumbens here and a tag of the caudate and the putamen. And to activate this region, we used a gambling task. Two patients get in the scanner, two cards are face down. They're asked to pick a card. If they pick a red card, they win $1, black card, lose $1. We rig the game. Everybody wins. Everybody gets paid to be in the scanner. They're thrilled. And this is what it looks like when you look at the sample as a whole in the win versus lose condition. You get very nice activation in this ventral striatal brain region. And this, I should mention, is the work of Lucille Caperon, a fellow Frenchman who was a postdoc in our laboratory a number of years ago. And what Lucille found when she compared patients who had received interferon alpha, in this case for hepatitis C, for 4 weeks, compared to control individuals who had hepatitis but were not receiving interferon alpha, you see significant decreases in activation in the win versus lose condition, right, left, and bilaterally in the ventral striatum. And this decreased ventral striatal activation that you see in these interferon alpha patients is then associated with reduced motivation in these individuals. You see quite a strong correlation with the sample as a whole. But if you look in the red dots here, they're clustered over here, which is low activation in the win versus lose condition in the MRI task, and high reduced motivation as measured by the multidimensional fatigue inventory. So 4 weeks exposure to an inflammatory stimuli leads to reduced ventral striatal activation in this basal ganglia region, and that's in turn associated with reduced motivation. Now, I'm not just me here yapping off about this effects of inflammation on the brain. This has been demonstrated in ultimately 3 laboratories now independently. In the UK, it's in California, endotoxin at UCLA, and typhoid vaccination in the UK. So work of Neil Harrison and Naomi Eisenberger, and they've shown administration of endotoxin or typhoid vaccination to healthy controls does exactly the same thing, reduces ventral striatal activation in association with depressive symptoms including reduced motivation. So 3 different inflammatory stimuli, 3 different laboratories, all finding the same thing. You could take this to the bank. This is what's called reproducibility, rigor and reproducibility, which is a huge thing in the US. Don't know about France, but these are the kind of reproducible findings that you can use as anchors to look at potential outcome variables in terms of an anti-inflammatory treatment or other types of treatment where you're targeting the effects of inflammation on the brain. So what might be leading to these changes that we're seeing in the ventral striatum at a neurotransmitter level? And in order to study this, we developed a rhesus monkey model of cytokine-induced depression, again, using interferon alpha. Interferon alpha makes monkeys depressed, just like with humans. And we were interested in looking at dopamine release in the striatum. Dopamine plays a very important role in reward processing and motivation in the striatal brain region. In order to study this, we used in vivo microdialysis. We put a probe into the region just north of this ventral striatal region. And these are awake, alert animals. And these probes both can sample extracellular fluid as well as put in various drugs that can stimulate dopamine release. And after 4 weeks of treatment with interferon alpha, what you see is dopamine is basically knocked down, knocked out in the interferon alpha treated animals. Same thing with homovanillic acid, which is the major metabolite of dopamine. And then if you go in to stimulate dopamine with either potassium or amphetamine, you get this very wimpy dopamine response. So you're seeing this decreased dopamine release in the ventral striatum, which is the same region where we saw the changes in the humans when we went in and stimulated with a hedonic reward task. And just like in the humans where we saw the ventral striatal changes map onto motivational deficits, in these animals, decreased release of dopamine was associated with decreased effort-based motivation in these animals. Reproducibility, is this something that happens in other model systems? The answer is yes. This is in rodents. This is in rats. This is the work of John Salomon from the University of Connecticut, United States. And he gave these animals IL-6 intraperitoneally and did in vivo microdialysis, much like we did, although he, instead of having one probe in the area, he put multiple probes in multiple different locations and focused on the nucleus accumbens, which is part of that ventral striatal region. And it really, these are the probe placements here. It doesn't really matter where he placed the probe. After administration of interleukin-6, you see this marked reduction in extracellular dopamine, and that decreased dopamine was associated with decreased effort-based motivation in these animals. So again, this is a reproducible finding and tells us there's something going on with dopamine that may underlie these effects that are occurring in the ventral striatum in our functional magnetic resonance imaging studies. This can be reversible. So this is a work of Jennifer Felger in our group. And what she did is she put levodopa into the brain of the monkeys and then did the, these were monkeys treated with interferon alpha or not. And here's levodopa. And administration of levodopa can completely reverse the effects of interferon alpha on dopamine release by amphetamine. So this can be reversed by using various drug therapy. And as I'll tell you in a minute, we're actually doing clinical trials using L-DOPA in patients with increased inflammation. So as we start to look at this path from inflammation to behavior, it's important to kind of understand, well, how does this signal get up into the brain and how does it get into this area, this nucleus accumbens and the striatum and the basal ganglia? And so we wanted to look to see what were happening to the immune cells. And these are postmortem samples from our rhesus animals who've been treated with interferon alpha or controls. And we're staining here for microglia and macrophages using an IBA1 stain that stains both of these cell types in green here. And in the control animals here, we're in the globus pallidus, which is a basal ganglia nuclei, and as well as in the striatum. And we're looking around the blood vessels and the meninges. And here in the brain parenchyma, not much going on in the control animals, but look what happens in interferon alpha treated animals. 4 weeks of interferon alpha, you're seeing activated microglia. Based on other studies that we've done, these microglia are producing chemokines that then draw macrophages from the circulation into perivascular regions and the meninges in the striatum, where they're poised to release inflammatory cytokines. Now, for us, this was all fine and well, but if they release cytokines, how does it get into the brain? Because the cytokines are too big to freely pass into the brain in this particular region. And so we were especially intrigued when we saw this study by Menard, who's here in France, as well as Scott Rousseau from Mount Sinai. They did a study in rodents, mice, where they exposed the animals to chronic social defeat stress and found that that leads to increased permeability of the blood-brain barrier to interleukin-6 in this very region of the nucleus accumbens that we've been talking about all along. Now, in this paradigm, what they do is they take a mouse, average mouse, and they put the mouse in a cage with a breeder mouse, a bully mouse, much larger, much more intimidating. And that mouse will sort of freak out the little mouse, as well as there's a little bit of snipping and a little biting and a little wounding. So it's a combination of both a psychological stress as well as a physiologic stress that can lead to inflammation. And then what they did is they injected these animals with a green labeled IL-6. So this is IL-6 labeled green with biotin. And then they looked throughout the brain to see if there were any differences in animals who become depressed in the context of this stress versus animals that are resilient to becoming depressed in the context of this chronic social defeat. And they found the biggest difference, in fact, the only difference they found was in the area of the nucleus accumbens. And what they saw in control animals was this is the blood-brain barrier here in red, nice intact blood-brain barrier, no IL-6 getting into the brain. These are the stress-resilient animals. They don't become depressed in the context of chronic social defeat. Nice blood-brain barrier, nice intact blood-brain barrier, no IL-6 in the brain. Look at these individuals, these animals that are stress-sensitive. The blood-brain barrier is in tatters here, and IL-6 is pouring into the brain, into this very region where we see the decreased dopamine production and release, and we see the changes in ventral striatal activation in the fMRI studies in the humans, all mapping on to decreased motivation. So we got a pathway that's kind of converging here with multiple different types of data that are telling us that inflammation is going after the basal ganglia. So based on this, this access of cytokines directly to dopaminergic neurons in the ventral striatum, and particularly the nucleus accumbens, we wanted to look at the direct effects of cytokines like IL-6 on dopaminergic neurons to, again, see if there are treatment targets that might be revealed here that might be used in clinical trials. And to do this studies, we've used induced pluripotent stem cells. These are human cells, iPSCs, and these can be grown into mini brains or 3D human brain organoids. There's a series of procedures that are used to convert fibroblasts or blood cells into these pluripotent stem cells, which then can be redifferentiated into any kind of neuronal system that you want. And we developed midbrain organoids, which if you do a cross-section of this day 84 organoid, you can kind of see the shape here, a little spheroid organoid here, and you cut it in down the middle, and you look inside and you see it's chock full with dopaminergic neurons, as well as in staining in green, these cell bodies, and then in red staining is tyrosine hydroxylase. These are the axons, and tyrosine hydroxylase is the rate-limiting enzyme in the synthesis of dopamine. We wanted to know whether these dopaminergic neurons express receptors for IL-6 and TNF. Here our tyrosine hydroxylase is in green, and you can see, yes, indeed, These cells express IL-6 and TNF receptors. These tend to be around the cell bodies here, but you do see some axonal expression as well in these cells. So the dopaminergic neurons express receptors for inflammatory cytokines. And what happens if you treat these organoids with interleukin-6? Fairly high dose for 24 hours. We treated them. This is a control condition here. And this is a high-performance liquid chromatography, which measures the levels of dopamine. In yellow here are just the standards to tell us where the different metabolites of dopamine are. And here's dopamine itself. Here's a nice peak that you see in the control organoids. And look what happens if you treat with IL-6. You almost completely obliterate the production of dopamine by these organoids, and there was no evidence of any cell death. So this is a physiologic phenomenon. And very briefly, what we wanted to see was, well, what exactly is inflammation doing to change this production of dopamine? And as I said, with chronic stress, you get an increase in blood-brain permeability, and in the cytokines go, and IL-6 binds to its receptor, and that receptor is tied to Janus kinase signaling pathways. A little difficult to see, but these are JAK pathways. And then there's a number of possibilities of what IL-6 could do. It can affect the synthesis, the release, the receptors, the reuptake. And so what we did is we treated these cells, the dopaminergic cells. These are 2D cultures now, and looked at the gene expression pathways that were affected in dopaminergic neurons as a function of IL-6 treatment, more physiologic concentrations here versus vehicle. These are the volcano plot. These are the downregulated genes. We were very surprised to find a very consistent series of pathways that were affected, and they all had to do with vesicular-mediated transport and release. So neurotransmitter release, packaging and release appears to be what's affected, and you can see the different pathways that are significantly downregulated: neurotransmitter transport, synaptic vesicle exocytosis, vesicle-mediated transport in the synapse. And so what we wanted to do is see if, well, can we use a drug to reverse these effects? And so this is a Janus kinase inhibitor, so it's inhibiting the pathways by which IL-6 does its thing, does the signaling. And so here we have IL-6 plus baricitinib. This is an FDA-approved drug for the treatment of rheumatoid arthritis, and it's also the only approved drug in the United States for the treatment of acute COVID. And so we compared IL-6 plus baricitinib to IL-6 alone, and now we're looking at the upregulated genes, and you can see that baricitinib has completely reversed the changes that we saw previously. And again, the pathways that are upregulated are all focused in on this area of synaptic transmission or transmitter transport and the function of the vesicles, recycling, regulation of dopamine secretion. So now we have a platform where we can not only understand what is the mechanism by inflammatory cytokines can influence dopamine production and release, but we also have a platform for drug discovery. And in fact, based on these data, we are now using baricitinib as one of our anti-inflammatory agents to treat patients with increased inflammation, in particular HIV patients at this point. So are there other targets in the brain that we can look at? Especially much of what I've talked about so far is where we are administering inflammatory stimuli to individuals and then looking at changes in what's going on in the brain. But what about patients who are depressed and have increased inflammation endogenously? Does this affect these dopamine pathways and circuitry in the brain? In order to study this, we've used resting state functional connectivity. I think most of you are familiar how this works. These are looking at relative oscillatory activities occurring in neurons and seeing whether neuronal systems are firing or active in concert, or whether they're out of sync. And those that are in sync, we believe, are talking to one another and represent circuits that then can be mapped onto behavior. And we were particularly interested in this reward circuit. These are the circuits that are involved in motivation as well as motor activity. And here we are again with the ventral striatum, and where we saw all the changes with the administration of inflammatory stimuli. And we wanted to see in depressed patients with increased inflammation, what happens to the connectivity of this brain region to the ventromedial prefrontal cortex, which is really the heart in red here of your reward circuit. And these more dorsal regions of the striatum are connecting up here with more motor regions. So this is motivation and motor activity that we're talking about in terms of looking at what the effects of inflammation are. And again, as I said, these are depressed patients. So to measure endogenous systemic inflammation in these patients, we used CRP, again, as I mentioned, produced by the liver, and thanks to the American Heart Association and the CDC, we know what is a low, medium, and high level of inflammation based on risk for cardiovascular disease. How these numbers were derived. So what we wanted to know, as your inflammation goes up, you're a depressed patient, as your inflammation goes up, what happens to the connectivity of the ventral striatum to other brain regions? So we did a whole brain analysis, and what we found, what we found was that this region that showed the biggest change as inflammation went up was a decreased connectivity to this ventral striatal region. So basically, as your inflammation goes up, your reward circuitry is tanking. And so you can see this here. This is a highly reproducible finding. We've shown this in several other, both animal models as well as in various human disorders. As your CRP goes up, connectivity between the ventral striatum and the ventromedial prefrontal cortex goes down. And that decreased connectivity that you see in the rewards and motivational circuits is associated with reduced motivation. And if you look in dorsal regions, it's associated with reduced motor activity or psychomotor retardation in these individuals. Can you reverse it? Well, Jennifer Felger was feeling frisky, and she figured, well, let's give them L-DOPA. And so this is pharmacologic fMRI. We do a baseline scan, then we we give the patients L-DOPA, and then we scan them again. We're not putting L-DOPA in the brain. They're taking it orally. These are depressed patients. And in these depressed patients, if you have a high CRP, you have reduced connectivity in the ventral striatum to the ventromedial prefrontal cortex. And so you can see this decreased connectivity under baseline conditions, and then you give them a dose of L-DOPA. Not much happens here in these individuals with lower CRP, but those with high CRP not only return to normal, but there's an overshoot suggesting there's supersensitivity in these pathways. And this increase in connectivity was associated with increased effort-based motivation in these individuals. So patients with high inflammation may preferentially respond to dopaminergic medications. So if you give them SSRIs, they don't respond, which is what I showed you at the very beginning, that these patients tend to be treatment resistant. And this may be part of it, is that we don't typically use dopaminergic therapies as frontline therapies in depression. And we currently have several clinical trials that are ongoing using drugs that are a little more dopaminergic in nature. And we're especially seeing some traction here with L-DOPA in treating patients. So dopamine is a target in patients with high inflammation as you begin to develop targeted treatments for this patient population. So what about inflammation? This is probably the lowest hanging fruit. And you're saying, well, why isn't he talking about blocking inflammation? And yes, we've done these studies. We continue to use these studies. We have used monoclonal antibodies to TNF to block TNF. We've used infliximab, and we did a trial to treat patients with treatment-resistant depression. We like these drugs. They're monoclonal antibodies, as I mentioned. They're very potent. They have very few off-target effects, limited drug-drug interactions, and we did a 12-week randomized controlled trial in this population. This is just a design of the study. We stratified based on sex and CRP, although we did not enrich for high CRP in this study. They got 3 infusions of infliximab, and then 30 per group, And then we studied them depression rating scales for 12 weeks. This just gives you a feel of the patient population. I mentioned about obesity. These patients were recruited for treatment resistance. So we didn't recruit them anything having to do with inflammation. We just said, are you treatment resistant? Come on in. And the people that came on in were obese and had very high levels of CRP. And if you do the math, where you look at the number of people with depression, the number of those who are treatment resistant, and the number of those who would be expected to have high inflammation, which was 50% in our patient group, that represents somewhere in the neighborhood of 5 million depressed patients, at least in the United States, who are treatment resistant and have high inflammation. You can compare that to those with rheumatoid arthritis and inflammatory bowel disease. That's a lot of patients. So we were extremely excited about this trial. And like many trials that have been looked at anti-inflammatory agents in depression, this is the result we found. Nada, nothing. There's nothing there. There's no difference between these groups whatsoever. Of course, in those days, we were thinking and we were hyping the fact that that depression was an inflammatory disorder, which, as I said, no, it's not, only in subgroups. So we asked the question, does it matter what your CRP is at baseline, whether or not you respond to an anti-inflammatory drug? And the answer was yes, that your response to treatment over time was dependent on your level of CRP at baseline. And the sweet spot was a CRP greater than 5, which was about a third of our sample. And you can see in individuals with a CRP greater than 5, there's almost a doubling of response in infliximab versus control. And look at the people with lower levels of inflammation. Actually, they do worse. So giving anti-inflammatory drugs, and there's some literature on this, giving anti-inflammatory drugs to people who are not terribly inflamed may not be a good thing. What symptoms got better? Here we are at baseline, and we're moving to 12 weeks. If we go in a negative direction, that means your symptoms are getting better. These are the 17 items on the Hamilton Depression Scale. You can probably guess that that's the anhedonia item, and it's really the winner here. But you also see significant decreases in psychomotor retardation. So that's that ventral and dorsal striatum that we're hitting and seeing effects in. And that's the anxiety circuit there that I talked briefly about before. Interesting, suicide also was significantly decreased, and there is a relationship between inflammation and suicide, an interesting sidelight. So basically, there are a number of, this is a very target-rich environment, going from inflammation itself to effects on dopamine and dopamine-related circuits. We didn't have time to talk about glutamate, but we've done a lot of work here. And then leading to specific symptoms that are tied to this pathway. And this really gives us a way to focus our efforts on how we go about designing clinical trials. So how do you overcome the trial failures? You enrich, you stratify for inflammation, high, low, however you want to do it. And you use biomarkers like CRP to determine who's inflamed and who's not. Then that overcomes this problem that in the literature previously. We talked about before, these non-specific measures of total symptom severity, which really is highly non-specific and not necessarily relevant to what inflammation does to the brain. So we need to tie our outcome variables to what inflammation does to the brain, look at reward circuitry and associated symptoms of anhedonia, also the anxiety circuitry and symptoms of anxiety, arousal, and alarm. And finally, most of the drugs off-target effects, we really need to use more specific drugs where we demonstrate that they're actually hitting the target. They're reducing inflammation either in the brain, ideally, or peripheral blood. And then our outcome variables relative to target engagement should also include potentially neurotransmitter systems in the brain or neurocircuits in the brain. So the time is right. We need to be smart about this. Selected patient populations focused on targets and treatments that are associated with the impact of inflammation on the brain. And I think this field, this area of the effects of inflammation on behavior and its role in psychiatric disorders can really lead the charge in precision medicine in psychiatry. So let me just thank all the people, a lot of people that do this kind of work and do this work with us, and the funding agencies that we use. And I want to thank all of you for listening to me in this presentation. So I'll stop there and thank you very much.
Thank you very much, Andy. It was really great talk. I'm sure all our dopamine level are high now because we're so excited by these results. Thank you to have all convinced us about the importance of immunopsychiatry and precision psychiatry. So we don't have lots of time because you have been just at the right time to present this data, but I have two questions in the chat and I also have two questions. So long questions that I'm going to try to summarize. The first one is asked by Bruno Auzerat from Bordeaux and the lab of Lucille Capuron. Quickly, so many thanks for this wonderful presentation. I have a basic question. Although obesity, overweight, childhood trauma, chronic inflammatory autoimmune disease, etc., commonly cause inflammation, the immune mechanism involved in the development of depression are presumably different. Perhaps the best strategy is probably to better identify immune signaling pathways that are more specifically implicated. What do you think about this critical issue for the promotion of targeted and tailored treatment strategies of inflammation-driven depression? You have an hour to answer.
Well, then I don't need to. Right now, if you want my honest opinion, I don't think it matters. Much of the the inflammatory response that occurs is an inflammatory response that can be contained by many of the anti-inflammatory drugs that we have. They tend, even if it's a JAK inhibitor or it's a blocker of TNF, it tends to bring inflammation down, regardless of whether it's rheumatoid arthritis or inflammatory bowel disease or eczema. These drugs have cross activity in terms of their ability to reduce inflammation. And we know for a fact that eczema, rheumatoid arthritis, inflammatory bowel disease have very different sources and mechanisms and pathways to their inflammation, but the diseases all respond relatively well to the same categories of drugs. So we don't have to get too fancy here, but I agree with you 100% that we need to know more about the immune signaling pathways and more about the immune system, and they won't fund me to do it because they point to that paper I showed you before and they say there's nothing there. Why should we be studying the immune system and its effects on the brain if we can't show that an anti-inflammatory drug makes you less depressed? And we can thank all the people who've done the studies before, which didn't take any of the things that I mentioned today into consideration. So how you do a trial matters, and it matters a lot.
Bruno, okay, you have your answer.
That's fine.
Fine. Okay.
That's fine.
Then we have.
I'm on the same page, Bruno. I'm with you.
We continue the discussion this summer in Bordeaux. I have another question.
Oh, that's right. Okay.
Yeah. I have another question from Marion Foisel, who is a PhD student in the lab. Thank you, Andy, for the rich and fascinating presentation. And she has a question about the timing of immunomodulatory treatment in depression and would like to know whether there is a right or a specific timing to prescribe immunomodulatory treatment to depressed patients.
I think that's one of that's a fabulous question and something I've been really struggling with, and thinking about where is the best place? I mean, is this something where we want to do like everybody else has done up to this point with any anti-inflammatory drug, that we take patients who are currently depressed and we do an 8-week trial of an anti-inflammatory agent? Is that what we want to be doing? Our cardiovascular colleagues are doing studies that go over years, and what they're looking at is how someone does over time. Now, is it that inflammation is a sort of a chronic insidious process that is driving behavior and leading to people having episodes of depression or precipitating episodes of depression? or is it the main player in a given episode? So I think we need to start thinking about whether we're giving chronic anti-inflammatory treatment and looking to see a better trajectory of the disease over time, like in cardiovascular disease, or should we be looking at the acute effects of an anti-inflammatory agent within the context of an episode of a disorder? And that I don't know the answer to, but I think we're ultimately going to need to do both, because the action may be in the chronic treatment and not in the acute.
She's also asking if a patient that has been suffering for depression for 20 years should be treated the same way as a first episode patient regarding to immunomodulation.
Yes, it seems to be the case. The anti-inflammatory drugs that are out on the market now, you may not get the kind of advertisements we get on television, but now they're advertising using these very strong JAK inhibitors for the treatment of eczema. And as a psychiatrist, I would say that depression is at least as serious as eczema, if not more so. I've not seen any patient die of eczema. I've seen a ton of people die from depression.
Now we have another question from Nicolas Marie, who is asking if IL-6 or TNF-alpha affect other neurotransmitter than dopamine.
Yes, they do. And that's been extensively studied in animal models. Not so much in humans. We got to dopamine. It's actually an interesting little side story. I'll tell it very briefly. Our first studies in monkeys, we did some brain scans that were just PET scans. We noticed basal ganglia were lighting up like a Christmas tree. And so something was going on in the basal ganglia. So we wanted to see what was going to go on if we gave this drug to monkeys. We had a monkey downstairs who had been given MPTP, which is a drug that's used to induce Parkinson's. The animal did not develop Parkinson's, and so the animal was, when given MPTP, so the animal was of no use to anyone. So this animal was just sitting there, and so we said, why don't we treat this animal with interferon alpha? We gave the animal an injection of interferon alpha, and with 24 hours, the monkey exhibited full-blown Parkinson's. So we had some very early ideas, very early on, that dopamine was being affected. But yes, it does affect other neurotransmitter system. And glutamate was, I can tell you a glutamate story too. So that was that, including glutamate, but other monoamines.
Thank you. And Nicola has another question, which is is CRP a good marker of inflammation in mice? And what would be the level that reflects a high inflammation in mice, I suppose?
No, I think in rodents, people like to use like haptoglobin or serum amyloid A. There's some other acute phase proteins that seem to be a little bit better in animals, but yes, rodents do have increases in CRP, and those CRP levels would track on to inflammation. I have no idea what the corresponding levels would be. I can, they're typically not, people don't use the turbidimetric assays, which are the ones that are used for humans. And so once you start doing ELISAs, God bless you. I don't know where you are on, because everybody's ELISA is run a little bit differently. There are lot to lot variation. It's why you can't use TNF and IL-6 that you measure with ELISA in a clinic. Not yet. They're just not reliable and reproducible enough.
Thank you. Andy, do you have time for 2 questions more?
Sure, absolutely.
So I have 2 questions. My first question is regarding selection or inclusion criteria of patients with inflammation in clinical trials or even in observational study. How to deal with all the comorbid elements like obesity, aging, stress, infections?
That's been something that's in the field, has kind of plagued the field a little bit, where people are saying, oh, well, you have to control, you got to control for BMI, you got to make sure that people aren't medically ill, and you can't have the patients be too old. Well, if you do that, you're basically ruling out all the people who have increased inflammation. So this is how people are coming to depression. And so I think that you can, I mean, we do this with cancer populations. People who are cancer survivors reliably have increased. They're more around 50% will show elevated levels of CRP or other inflammatory markers. So I think you can select on that basis. The obesity is a little bit different, because what we found, and I think Lucille has similar data, that once you get to a certain level of obesity, things start to get wonky. So if you get your BMI starts to approach like 45, 50, things start to flip. And we're not really sure why that happens. So yes, I think that it is certainly, if you're interested in inflammation and you're trying to do a research study and your CRP needs to be over 3, you might want to recruit heavier, older people. But we try to stay away from people with uncontrolled diseases, and we try to stay away from autoimmune disease, because that's maybe a little bit of a different animal.
And one last long question. Anhedonia is a key question. So you have alluded to loss of motivation, loss of pleasure, motor retardation. So how would you target really what's correlated to inflammation in this area? It's really a big question for us.
Yeah, I think it's a huge question, and I think it's where a lot of the action is, because there are people that do well with antidepressants but continue to experience anhedonia and continue to experience issues with motivation. And much of what we've seen is effort-based motivation, the willingness to expend effort for reward. This has been seen in the animal models, where the animals, if you just give them a treat, they will eat it, they will consume it, not a problem. But if they have to work for it, no way. We saw this in the monkeys. It's been shown in the rodent models. And so there seems to be something specific about that. We are on the verge of publishing some data on this as well, with our infliximab study showing that effort-based motivation does respond to anti-inflammatory treatments. So I think that, and thank goodness there are measures that the NIH has seemed to gravitate to. So the effort task is a task that people use in this area. And if you look at most of the motivational scales, or even the fatigue scales, things like fatigue, things like pleasure, things like the effort-based motivation, they're all conflated and all confounded. So it's very hard with the scales that we have to tease out what is loss of pleasure, what is loss of effort-based motivation. And most of, there was a paper that was by Diego Pizzagalli that was in the American Journal, the Green Journal, talking about mechanisms of anhedonia. And he defined anhedonia in the first sentence of the paper, where he said anhedonia is the loss of pleasure. And so there's no mention whatsoever of inflammation in the entire article, because it's not about a loss of pleasure. It's about a loss of motivation and a loss of effort-based motivation in particular. So these are concepts that, people are very interested in and parsing these things out. And I think there's going to be some nuance in how inflammation impacts motivation that's different than just the kind of garden variety of anhedonia around loss of pleasure.
Well, I think we unfortunately need to stop here. I want really to thank you very, very much for sharing all this extraordinarily important data with us. And we look forward to next discussion with you. Thank you very much.
Thank you. Bye, y'all.